Dr. Tushar Ranjan. He is assistant professor cum scientist in Department of Molecular Genetics and Engineering in Bhagalpur University. That is not Bihar Agriculture University, Bhagalpur. Uh, his research work includes understanding the mechanism of genome packaging and translocation in plant viruses and its identification and characterization. Apart from his scientific work, he is having a very enriched scientific lab with diverse biological sciences areas, which includes bioinformatics in his lab, molecular genetics, recombinant DNA technology, biochemistry, and RNAi. So he is having a very good lab to work with. Apart from this, he is having a very intellectual personality because he has been author of several international publications. He has published more than 10 papers in international journals, and he has been author of 10 books as well. And he has written 6 practical manuals in the books. He has been editor for several books as well. Apart from all this, uh, he has been awarded uh, from the DST SERB for a startup research grant and early career research from DST. And I would like to mention that Dr. Tushar has been graduated from College Patna. So this is a proud moment for all of us to listen from him. Now I would like to hand over the mic to Dr. Tushar. I would like to welcome to Dr. Tushar to present his presentation.
Thank you. Thank you, Rajesh. Thank you. Thank you, Rajesh, sir, very much. So, a very good morning to one and all. So, I'm Dr. Tushar. So, actually, first of all, I would like to thank Sanjeev, sir, for providing me the opportunity here. Thank you very much, sir. And I hope our previous speaker, they have covered all of the like various entrance examination to enter for PG courses like IIT JAM, ISC, NCBS, TF, JNU, and other things like that in detail. And they must have discussed about their strategy, how to crack those exams, and how to how to get placed in a very good renowned institute of India. So now, in today's lecture, I will take you all one step forward, like uh, what you will do after your masters, what you will do after your PG or after your MSc or MTech or everything. So what will be the next? So our whole today's talk, they will be we will be focusing only on these aspects. So how to move ahead and how to gear up for the research and higher studies, basically. So, I will just brief about myself. Basically, I'm from Bihar, and basically from a very undeveloped district. I would say it's Supaul district. It's the Supaul district from the Bihar. So in terms of, if we talk about in terms of development and other connectivity and also it's still, I would say, it's an unprivileged area if we talk about in terms of Bihar and all. And I did all of my till 10th and till +2. I did all kind of schooling in my village itself. So I can say I have nothing to hide anything. Actually, I have almost up to 4th or 5th. I have studied on, we used to take our Bora along with us, and then we used to sit over there and something. It's what completely pure, pure government school. It was not like something, some deep, some JBS and something like that. It was not like that. So from there, I started my journey. Then I did my intermediate. Then then definitely without wasting our time, I just entered into this biotechnology for BSc biotechnology in College Patna. So their role of Sanjeevni sir comes into the picture, because that time I was completely naive. I was completely blank because I had no idea like what to do. I thought like I did my intermediate. My parents, they wanted me to go for, like other parents, they wanted me to go for medical preparation and other things. But because of something, my personal issue, I couldn't pursue for that thing. But without wasting my time for medical and other things, I decided to opt for this biotechnology stuff, because that time in 2007 or 6, it was like biotechnology. People were saying like biotechnology. So days are coming, so biotechnology will be get their own boom or something like that. So we decided for go for that. I entered into N college for BSc biotechnology, and my batch was between 2006 to 2009. So then I entered over there. I thought, because there were one thing that kept on motivating me. Like whenever I used to see some some journals, some things, they have written like Smith et al. Has said that according to this theory, something like that. So this one has gave this theory. This one has said something like that. If when I was going through some books and all, so at that time I got fascinated. So can we do something? Can we go for that far, so that I will be able to, I will be on that position, so that I can give some concept, I can give some mechanism, I can give some fundas, so that people will follow all over the world. So those things were in my mind, but I was not confident enough. Like our previous speaker Sanjeev sir said, it's one of the demerits for Bihari student, you can say. So we always underestimate ourselves. So that time, of course, I would say I underestimated myself. Then once I enter into AN College Patna, then I would say I would definitely, sir, in front of here only, I would like to acknowledge, sir. Thank you very much, Sanjeev sir, for uh, your support and for your guidance, I would say. Because that time when I entered there, so that was, as I said, I was completely naive and I was completely blank what to do, how to pursue for higher study, because higher study was always fascinating for me. So I always want to go for higher study, like PhD from some good institute, postdoc, then explore the world, then again you do some teaching and research, something like that. So I will tell you one thing, like for the first time, he asked me, because that time he was the teacher, and when we attended his class, so he used to say, you read, if you definitely want to pursue a career in biotechnology, then you have to follow this book, that book, and like that, so many things he was saying. So then I contacted him, and I went to his office, and I asked, sir, I'm very much interested in this area, biotechnology, and I want to pursue my career, and I want to explore myself in biotechnology field. Then he said, it's okay, don't worry. If you come here, so I will just guide you what to do, what not to do, and how to, we discuss like strategy, how to read what book, and how we shall prepare ourselves for to crack those exams like IT Jam, JNU, and then CSI, JRF Net, something like that. So these are the exams. So what happened? So that time only, if I remember, that time he suggested to go for some good books. Good books, bole to, like that time I came to know about a few of the books, like Le Ninja, biochemistry. If we talk about the biochemistry, there is one book, Le Ninja, White and White. If we talk about cell biology, there is a book, Gerald Karp and Albert, and other books like that. If we talk about this molecular biology, we have Watson. If we talk about this genetic engineering, gene manipulation, we have Primrose, and we have T.A. Brown, something like that. And if we talk about immunology, we have QA. So during my graduation only, I covered all those books, like Le Ninja, because he asked me, if you really want to if you want to pursue your career in this field, definitely you have to follow this book, and you have to you have to read thoroughly these books and all. And I followed him blindly. I'm to be honest, I followed him blindly, because that time I didn't have any idea. No one was there to guide us, to lead us, like kahan jana hai, kaise jana hai. No one was there. So I followed him blindly, and whatever he gave the name of the books and all, I started reading all those books and all. I studied all those books. I cracked the I cracked the exams. Finally, I got the place. You know, why doing my master's program, again same thing you have to study, like again like cell biology, molecular biology, biochemistry, genetic engineering, immunology, plant biotechnology, animal biotechnology. These things only you have to study again in detail in during your masters and PG and all. So that time, my when I was there, and I was the first when I remember still. So there was my teacher. Her name was Reenu, and she was saying like, uh, "Did you ever heard name of the book Chube for immunology?" I said, "Yes, ma'am. I heard that name of the book, and I have already studied those. I have already studied once. I have already completed this whole book once." So she was very surprised. Means BSc level पर ही लोग ये सब book पढ़ के आ रहे हैं. Then she asked, "From which state you belongs to?" I said, "I'm from Bihar." So, "Is it so?" Like, "Yes." "I thought you must be from Kolkata. You must be from some other metropolitan city and something like that." So I said, "No, ma'am, I'm from Bihar, and I have studied all those books." So that's what I'm telling you. So first of all, uh, I would like to say, language is not a barrier. ठीक है? You don't have to be very, very influent in English to cracking these exams like CSIR, NET, and these exams. You have to be confident enough. If you know the things, if you know the basic fundas, if you know the concepts, if you know the basic things, then I'm damn sure no one will stop you. चाहे वो interview हो, चाहे कुछ भी हो, no one will stop you. So for me, my friends, you don't have to be, you don't have to be very fantastic in, spoken English. And, yeah, of course, it is required for improving your personality. And, it will increase day by day. So when I was in village, that time, so I'm telling you, till graduation, I gave all of the exam in Hindi medium. And now I can write the article, I can speak fluently in English. So all I would say, this thing will come, gradually. It will not come suddenly in a night. जैसा ऐसा नहीं कि आज हमने देखा और कल सुबह आप fluent बोलने लगोगे इंग्लिश. So it will take time, and these things comes gradually actually. So most important thing is to have faith in you, and yes, be confident. So I don't think interviewers, they will ask you to know always. I still remember when I was attending one of my interview in NCBS Bangalore. So again, like Sandeep sir said, like as a Bihari student, I was like, I was very nervous. I was seeing people around me, they were talking in English. Oh dude, come on, what sir, like this, blah blah. They were talking, and I was, I was all alone over there from the Bihar, and I was sitting one side in the corner. Then I said, क्या फर्क पड़ता है यार, चलते हैं अंदर, चलते हैं, जो होगा देखा जाएगा. Then I entered in this that room, and I still remember there was a Surolia, and he was asking the question like, "So Tushar, from where you are coming?" I said, "I'm from Bihar." "Oh, you are from Bihar." So then, he automatically he started speaking, started taking my interview in Hindi. But I said, "No, sir, I'm also very much comfortable in English, and I think we shall start our conversation in English also." So that's what I wanted to say. So language is not a barrier at all, until unless you knows the funda, until unless you knows the concept. So without taking too much time on this, I would like to start my this session. Piyush, Rajiv sir?
Yeah, yeah.
Can you
I
share my
was
slide?
just saying that I have downloaded, your slides are ready.
Yeah, please.
So, no, you continue. You continue first. It was very, very interesting story, in fact. Then we'll start.
I will connect my story somewhere in the mid. So
Okay,
you just
fine. This is what
the
happens with
slides.
every Bihari students, in fact, when we come out of
Yeah,
Bihar in different states.
yeah.
So,
So there should not be. That's what I'm saying. It's not a language, not a bad. Like we can see Pius
exactly.
and Pawan. They are, they are leading all over the world. So you can see they are
Correct.
also Bihari and really doing excel in their field. So and these things, I would say, thanks to Sanjeev sir, who motivated us to reach up to this goal.
Exactly.
I would like to thank Sanjeev sir for all those things. So today mostly we will be covering, like as I said, जैसे हम लोग बिहार से हैं, like जैसे आप एमएससी करते रहते हो, पेरेंट्स पूछने लगते हैं कि बेटे कब तक पढ़ाई करोगे, कब तक पढ़ाई करोगे. It's enough time. No, it's time to अर्न. अब आपकी फैमिली को भी आपको ध्यान देना है. So that's what I'm telling you. If you crack some of the good exam like for masters like IIT JAM, JNU, and ISC, NCVS, TIFR, definitely you will get the scholarship for that. And I think this must have covered by our previous speaker on previous Sunday. So today we'll discuss about like what are those fellowship that would help you for pursuing PhD. Because see, simply getting PhD degree from some institute and doing a quality PhD, both are two different things. Both are completely two different things. So I would like to say that better you qualify some of the exams, like there are a few this government agency like your CSIR, DBT, ICMR, GATE. They provide the fellowship to pursue your higher study, to pursue your PhD from some good institute or some well-known institute. So this is all about, I was saying, like this success, actually it's a journey, it's not a destination. So I've covered all those things. So we should I don't want to take much time on this, so I directly want to come to my presentation or my talk. Next slide, please. Yeah, so these are the different fellowship for pursuing PhD. Like first is your CSIR, CSIR. Then there is a DBT. Then there is a ICMR. I'm not able to see complete screen, Rajiv sir. Yeah, no, it's okay. Can you just hide this? There is one bar over there. You just hide over there. So yeah, now it's visible for me. So then fourth one is a GATE. Next slide, please. So now first I will discuss about these. I will be focusing these CSIR, how to crack those exams, what should be our strategy, and how much we should spend time on which portion, something like that. So I will basically focus on the CSR part. But yeah, it's obvious that once you have thoroughly studied the syllabus of CSIR, then no one can stop you in cracking exams for DBT, ICMR, GATE, and all. So that's why for taking this CSIR as a prototype, so I will discuss all the syllabus in detail for this cracking this CSIR JRF net. So this is an opportunity for higher studies, basically what we call research. So we have like CSIR JRF conduct, they provide the fellowship for other fields also. So basically I will be focusing on this life sciences because we are from the life sciences background. So this, we all know that this CSR UGC provides the awards of JRF, and they also give the certificate for eligibility of lectureship as well. And if you talk about the timing, they provide 3 hours time, and they are total of marks. If we see, they are of 200. So in our time, not our time, I would say before 2011 or 2010. So we used to have, this CSIR exam in 2 sessions, first half and second half. It used to be a very long and exhaustive session. So in first half, we have to, it used to be completely objective based question, and in second half, there used to be a subjective one. So we have to write the answer in that particular space provided, something like that. But now these days it's not like that. So this procedure has been completely changed, and these exams, they complete this test, they are made up of only multiple choice question, and there is only single paper test, and they are having multiple choice questions. So these complete question paper, they are divided in the 3 parts. And there is one more thing that I would like to mention over here. So there is a negative marking for if you attempt some wrong answer. So there will be a deduction of 0.25. Means if you have done by mistake, if you have done some mistake for one question, so 0.25 mark will be deducted. So something like that. So next slide, please. So yeah, This is so. I was saying that the whole test, they are comprising of like three parts, like part A, part B, and part C. So here strategy comes into the picture. Like for the part A, so part A is basically they just want to know like basically your IQ level and like your like common sense, which kind of common sense you have. Like it's a completely normal research aptitude, you can say general science or some aptitude test or something like that. So here they will ask in part one, there will be 20 questions, okay? And candidates shall, they are required to answer only 50 questions, okay? And we all know that each question shall be of 2 marks. So अगर हम लोग देखें तो the total marks allocated in this part A shall be all around 30 out of 200. So there will be a 30, means total mark is 30 because we have to attempt only 15 questions. Okay? And there will be a 20 question. So, you know, I will just, although I will discuss these things in coming slide, but I just want to give over here some glimpse of the question, which type of question they usually ask in this part A. Like, you know, I have given the picture of here in my presentation, like dice, like just हम लोग ludo खेलते रहते थे. So like, what is the probability of getting a number 5? You know, very basic question, like very basic question they are going to ask, from the probability, from the permutation, from the combination, from you can say from basic biology, basic chemistry, basic physics. They will ask the question from all over the life sciences. But I'm telling you that for this part A region, you don't have to, you know, follow some, you don't have to read some book separately for these part, because you know these are the things they will ask the things that you face daily on regular basis in your daily life or something like that. So they will ask the question from those things only. So don't be panic for this. They will ask very basic and very concept based and very, you can say, IQ based question in this part A. And you will have to attempt maximum of 15 questions from this area. And this will comprise of total 30 marks. Now coming to the part B, it's the MCQ based part. So there will be again multiple choice questions in this part B, and you can attempt up to 35 question in this part B also. And like part A is in this part B, each question shall be of 2 marks. So by doing so, if there are 35 questions and each question carries 2 marks, so by doing so, if you can see, so there are 30 marks that need to be covered in this out of 200 for this part B. So 70 plus 30, then there is 100. So now remaining 100 is remaining. So this 100 is remaining completely for your part C. So I will tell you one more thing for this part B region. This part B, they will ask question from, you know, your +2 background. Like +2 background bole to, like your physics, chemistry, biology. Basically, you don't have to, you know, take taboo and make taboo about physics for this CSIR exam. They will ask very basic question from the physics. So you don't have to get panic for this physics. No, I need to study SC Verma. I need to study some very good book for physics and all something like. No, if you have studied, अगर आपने +2 level पे अगर आपने justice करा है अपनी पढ़ाई के साथ, if you have studied your +2 your physics, chemistry and math wala parts, then you need not to worry for this +2 wala portion for this CSIR things and all, because these questions are basically asked from this +2 wala area, like 10th plus 11th and 12th, basically intermediate region. And they will also ask few questions from the higher studies like cell biology, biochemistry, molecular biology. But this will be like simple objective, like objective based or some simple MCQ based question. Now coming to the part C, so here these questions, they will be of an analytical nature. Means they just want to test your research temperament, means your concepts. Means ऐसा तो नहीं है कि directly you have mugged up and you have appeared for exam. Because in part B, if the student who has completely mugged up the whole syllabus, they can still qualify part B and somehow part A. But part C, here your higher studies and things that comes into the picture. So here you have to be very paranoid about your question, because you know one question they carry, they are of 4 marks. So it means that during before 2012 and all. So these part C, they used to be, you know, completely subjective based. So now this time they have just converted them into MCQ, but you know, answer I would still say it's a complete. I would still say it's a complete subjective type, because you know they will provide some space in the box, and you have to do some calculation. You have to design something, and there will be space providing for this all kind of calculation and all kind of rough work and all. So in this region, you will have to answer any 25 question, and each question carries 4 marks, so 100 marks, so total 200 marks. So basically, the question that will be asked in part C, they are to test your scientific temperament, scientific concepts, scientific funda, something like that. And here, like all of the whatever books, I will discuss those things in coming slide. Next slide, please, Rajiv sir. Yeah, this is the syllabus of CSIR NET JRF. You know, there are 12 to 13 points are over there, but you know, you don't have to. I would still say you have to be, you know, you have to play smartly so that you can, you know, you can crack the exam. So I will tell you, and I will tell you all the mantras and all the strategy, how you will opt for this strategy, and how this strategy will help you in qualifying this exam. So I have given the snap of few of the books like Leninger. You know, I used to say in my here also, when I used to take class for graduation student and masters and PhD students. So if they are from, if I used to tell them, if you really want to pursue your career, then definitely you should have, you know, this book, this Leninger book in your hand, you know, because I would like to say, I just used to tell them, like, it's a, you can say it's a Bhagavad Gita, it's a Quran, it's a Bible for all of us, for all the students. So, you know, you have to be, especially your chapter number 1 to 12. They are basically this chapter number 1 to 12. They basically covers all kind of these biomolecules and this plasma membrane and other basic fundas. So you know, you just read all those chapters from 1 to 12. They are very helpful. They have covered all of the biomolecules. And you know, once you have see, there is no shortcut for this. You know, some people will say no. There are few instant notes. You just go for that, and and you will crack this exam and all. So just it's not just about, it's not just to crack the exam. It's not just to, you know, just crack the CSR and just enter into some institution for PhD. Means you should have, you know, you should know how to, because you know, from MSc to Masters to PhD. So now it's in PhD, you are not going to study anything. You are going to research. You are going to find. You are going to work on some novel aspects or novel findings. And at the end of the PhD, you will definitely you are going to give some novel findings to scientific community and all. So you know, by reading these books, you know, your research skill that will, you know, that will gradually develop day by day. So that's why reading the book, not only reading, means going thoroughly to these books that are very important. So I'm just naming few of the books that will be helpful in cracking these, not only cracking in these exams, this will also, you know, that will make you ready for, you know, that will gear up and make you ready for your research and higher study. So as I already said, for biochemistry, there is a Bhagavad Gita and Quran and Bible for us, like Leninger. So you have to follow this book. You know, in this book, they do not consider only biochemistry. If you go chapter 22 or chapter 23 onwards, there is a molecular biology portion also. So still you can, you know, you can read molecular biology portion in this book also. So this Leninger, they covers biochemistry, your chemistry behind your molecular biology, like all kind of replication, transcription, translation. What are those? Chemistry takes place behind replication, transcription, translation, and gene regulation and other things. And apart from that, the mid portion of the Leninger, like between chapter 12 to, you can say, 21 or 90th or 21st or something like that. In these region, you have like metabolism, complete metabolism part, like your glycolytic pathway, ETS, Krebs cycle, all kind of metabolic pathway they are discussed in detail. So These book itself, that's why I used to say it's a complete Bhagavad Gita, it's a completely Quran and Bible for us. So don't forget to read these books in details. I know in initial day, when I was in graduation, so when that time Sanjeev sir told me, Tushar, you just buy this book Le Ninja. So I went to, I will tell you one thing, because then everyone has, in our student life, everyone has financial crunches. So that time I said, sir, I cannot buy these original books, it's too costly, it's 5,000, 8,000, 10,000, something like that. He said, no, you go to the Patna Gandhi Maidan and Patna, that Ashok Rajpath wala area, and you will find the book in 1500 or something like that. So that time for the first time I bought this book. So first time I bought in my life, I bought that book was Le Ninja. So I bought that book and it cost, I think it was of 1200 or something like that. So I bought this book from this Gandhi Maidan and Ashok Rajpath wala area, and I bought this book. So not only this book, so this book was all about for biochemistry. But if we see molecular biology, so there is a book for Watson. So my friend, I will definitely suggest you to go through this. For molecular biology, you should definitely read these books. Reading the books, still I would say, in my batch, there were 45%. Sanjeev sir is knowing about all those things. So there were 40 to 45 students. But there were few 4 to 5 students. They moved forward to, they said they took the strong decision, and they said, sir, I want to pursue my career in the CSIR, in biotechnology field and all. So that's what I will tell you. जब तक ये आपके अंदर से ना आए कि नहीं, I want to pursue my career in biotechnology, I want to do something in research, I want to make career, I want to do excel in this science, I want to explore the world, then definitely then you come to this field, because there is no, of course there is for not even a single course there is a shortcut. So I would say there is no shortcut at all for this. If you really want to do excel, then you will have to read these books, you have to crack these CSIR exams, you have to enter into some good institutes. Then after finishing your PhD, then you can go for postdoc abroad. Then you can explore the world. Then you can come back again India. If you don't wish to come back India again, there is a lot of scope in abroad also. So that's what I used to say. So sky is your limit. So that's what I'm telling you. Once you enter into PhD, why we are saying that you should enter into some good institute? Why we are emphasizing on that particular statement? Good institute. People can do PhD from from, I don't want to take a name of the university. They can pursue. They can complete their PhD from some of the state university of Bihar, some of the state university of other university. But if you do PhD from here, and if you do PhD from some good institute like ISC, like IITs, like NCBS, like TIFR, like CCMB, then you can differentiate, and then you will come to know why we are emphasizing and we are insisting you to crack these exam and enter into those institute of higher reputes. Because once you enter to those institution, then you don't need the help of Sanjeevani sir, you need not the help of Tushar, and you need not the help of Sandeep sir and Rajiv sir and Piyush sir and Pawan sir. No one. Because I want to say once you enter to those university now, so you will not once you enter in that environment, you will start thinking by your own. So what's the next? So I have come up to here. Then you will see people working in the lab, people they are talking science, people they are enjoying science, they are exploring science. Then then आप completely एक दूसरे वे में चले जाओगे. Then आप खुद ही बोलोगे नहीं यार ऐसा कुछ नहीं है कि तुरंत जॉब, and यार people say कि नहीं इसमें जॉब नहीं है and all. No, मैं completely इस चीज को deny करता हूँ. See, at the age of 26 or 27, I got I did my master's, I did my PhD, I have few experience of 6-month experience of this postdoctoral also. Then I come back again here and I completed my, so I got the position for assistant professor. And right now I've got the fund, I'm doing the research, I have my own lab, I have my own students, they are doing fantastic research, and they are doing the good basic research and all. So it's, if anyone say कि नहीं यार इसमें कोई scope नहीं है, तो बोलना come on dude, क्या बोल रहा है, तो कुछ भी बोलता है, ऐसी कोई बात नहीं है. If there is a will बोला जाता है ना, जहाँ आपकी चाह है वही राह है, हिंदी में हम लोग बोलते हैं. तो अगर if you have a will, then definitely you will find a way. So इसीलिए मैं बोलूंगा, लेकिन there should be no, because I have seen one of my friends, I don't want to name over here, so one of few of my friends, Sanjeev sir must be knowing that किसके बारे में मैं बात कर रहा हूँ. So you know they were like they were keeping their leg on two boats. आप दो नाव की सवारी एक साथ नहीं कर सकते हो, राइट? आपको डिसीजन लेना है कि आपको या तो हायर स्टडी को जाना है या फिर आपको इस फील्ड को छोड़ना ही है, समथिंग लाइक दैट. So that's what I want to do. You have to be highly focused. You have to be, you know, first you set your aim. नहीं मुझे सब कुछ भूल करके कि नहीं मुझे जाना है इस फील्ड में, देन ओनली आप उसको जस्टिफाई कर पाओगे. अगर आपके दिमाग में रहा नो मास्टर करते करते नहीं, आई हैव टू, यू नो, आई हैव टू start working, I have to earn, I have to take care of my parents, I have to take care of my family. So still I would say yes, you will get the JRF, you know, in this CS, once you crack these CSR things and all, you get the, you know, 35,000 per month. So it's for us, for a student, it's a handsome scholarship. So you can, you know, you can take care of yourself, and meanwhile you can help your family also. That's what we did, that's what we all have done, that's what I did. So इस चीज के लिए इतना वरी नहीं होना है. बट हाँ, इसलिए मैं आप लोगों को बार बार बोल रहा हूँ, यू हैव टू फिक्स द गोल. नहीं भाई मुझे करना है, मुझे हायर स्टडीज को जाना है, देन आपके पास वो जुनून होने चाहिए, देन ओनली यू कैन डू दैट. अदरवाइज यू नो समवेयर बीच में ही आप, यू नो, यू विल फील टायर्ड एंड यू विल बी बैक, एंड आप कहीं और चले जाओगे, कहीं दूसरे चीज में अपने करियर को ऑप्ट करने जाओगे. सो दिस इज व्हाट आई वांटेड टू से. सो दीज आर द फ्यू ऑफ द बुक्स लाइक दैट मोलिकुलर बायोलॉजी. Then for genetic engineering and gene cloning, you have to study like T.A. Brown. There is a book for this genetic engineering. They have discussed all basic funda of gene cloning and everything. So, you know, I have all those books in my my own library of my own lab. You know, I use my students, my JRF or my MSc students, they used to come. I ask them, if you really, you know, I never force anyone. If they say, you know, a few of the students, they say, sir, I just want to get my MSc degree and I want to go for banking sectors and all. You know, I never forced them कि नहीं भाई तेरे को करना है, ऐसा मैं कभी किसी को नहीं बोलता हूँ. वो तो ठीक है, इट्स ओके, इट्स अप टू यू, बिकॉज कोई जॉब कोई खराब नहीं होता है. सो इट्स अप टू यू फ्रेंड, डिसाइड व्हाट टू डू. अगर आपको जहां भी जाना है आप जाओ, लेकिन यू नो बी, क्या बोलते हैं उसको, be honest with your, you know, your decision, be honest with your decision, be honest with your aim, then only you will achieve whatever you dreamed for. So these are the few things. I will मैं इन चीजों को connect करते रहूंगा धीरे धीरे. Then there is a book for the cell biology. There is a book Albert, you can follow that. Then of course there is a very famous well-known book for immunology, QA. Then there is a book for microbiology, Pelcha. There is a book for then of course book for, we have techniques, Wilson and Walker. These are the books. So you know, these 1 to 12. So like if we see 1, this is completely biochemistry. 2 is also almost, it's a cell biology and biochemistry. 3rd one is completely molecular biology. 4th one is again cell biology. 5th one is developmental biology. I will tell you the tricks about the developmental biology also in coming slides. And you know, if we see diversity of life form, ecological principle, evolution and behavior, something like that. These 3 things, and inheritance biology. So this is inheritance biology. It means it's your classical genetics. So you know, whatever you have studied for your +2 time. So for, you know, you will feel, I don't know how will you feel, but I'm going to tell you that for these 8, 9, 10th and 11th, योर एनसीईआरटी जो भी आपका +2 और 11th and 12th लेवल का जो भी एनसीईआरटी है, फिजिक्स, केमिस्ट्री एंड बायोलॉजी, जो भी बायोलॉजी के दोनों वर्गों में, इट्स मोर देन एनफ टू कवर दिस बुलेट पॉइंट नंबर 8, 9, 10 and 11. Next slide please, Rajiv sir. Yeah, so let's discuss about this first point, like molecules and their interaction relevant to biology. So these are the, you know, these are the subsections of this syllabus, like structure, like you know, that's what I used to talk. Like I have written over here, it's a Bible, Gita, Quran and Bible for biotech students. So I have given the name of 2 books, like, you know, Leninger, and one book is What and What. You know, what I have seen, there was one of my batchie, he was, you know, he was like very confused. Not he, she was very confused. She was like, Tushar, I will read this What and What also. I will read this Leninger also. And there is another book for biochemistry, Hooper. So I will read for that book also. I used to deny her कि यार ऐसा मत करो. Just follow one book. चाहे अगर तुम्हें लगता है कि what and what ही पढ़ना है, it's also a very good and this kind of book. So you can follow that book, but don't, so here I would like to, insist or emphasize or to this point. So just follow one book, चाहे वो what and what हो, चाहे Leninger हो. But अगर आप मेरी बात मानोगे तो definitely I will suggest कि no, you just go for Leninger. And there are many books, good books like What and White and Hooper, but you don't have to, you don't have to read the complete book, because Hooper is completely for medical biochemistry portion, and this What and What they have covered molecular biology part in much more detail and depth. So I would suggest for this this complete whole this syllabus, whatever it is showing on your screen, just follow the Leninger book for this complete. You will find each and everything in this. Chapter 2, this structure of atom, molecules, chemical bond. In chapter 2, there is a chapter 2 and chapter 3 of Leninger. So chapter 2 is completely water. They have discussed everything in detail in water, what chemical bonds and all type of interactions. So you must not you should read these chapters, because people they just ignore, this is the water wala, isme to most of the thing we know about it, hum log sab kuch jante hain, usko padhne ki zarurat nahi hai. No, I would like to say that no, first of all, before you start the Leninger, you start from that chapter number 1, chapter number 2, that water things. Then then you will get all the basic fundas, basic ideas, and onto that you can build, you can, it will be really helpful in studying the next chapter and all. Then of course these biomolecules like carbohydrate, lipid, protein, nucleic acid, vitamins. Then there will be all kind of stabilizing interactions, like all kind of buffers, bioenergetics, like all kind of metabolism, like glycolytic pathway, ETS, your Krebs cycles and other cycles. And then of course enzyme kinetics. Then your protein folding, protein conformation. Then your metabolism of this carbohydrate, lipid, amino acid, nucleic acid, vitamin. Here in this J wala portion, like metabolism of nucleotide, vitamins, and amino acids. So you don't have to worry about this metabolism of amino acid and nucleotides. CSR, very rare, CSR, DBT, ICMR, GATE, very rarely they ask the question from the metabolism of amino acid and metabolism of nucleotide. But you should have you should have basic idea. So how it was like de novo pathway, salvage pathway of this nucleotide biosynthesis. So you should know the basic things, but you can skip few of the things, something like. But in these things, so this complete whole syllabus, they are well covered by your book, what we call Leninger. Next slide, please, sir. So I will give you the Sanjeev sir asked me to give some sample question. So which kind of question is actually usually asked from this portion, from the biochemistry? So in 2018 they have asked one question, proton hooping, and they have asked. So, the beauty of CSIR, they will not directly ask question, and you will not get direct answer for that. So then how you will be able to answer that? So this is what I insist you all to read the books. So once you read the good text, once you prefer the textbook, then definitely you will be able to answer all those questions. So these proton hoopings, I have mentioned 3-4 questions. These questions have directly taken up from the Leninger book or any of the biochemistry good book. You can say White and White and Leninger, something like that. So this is they will ask something like, they will draw some pictorial, they will give you the pictorial representation. You can see over here this proton hooping. I have written over there, this is a proton hooping. So it's something like there are 11 players. There is a ball in first player. They pass the ball to second player. Second pass to third to fourth, and finally it reaches to the end of the last player. So this is what we call a proton hooping. In water, if we see the chemical structure of water, they do exist in hydronium ion form. S3O plus, what we call. You can see over here in the slide. So I cannot use the cursor. So you can see over here like S3O plus. Why we call them S3O plus? Because you can see O and there are 3 H. So that's why we call them S3O plus. Because water, they do exist in this. They follow this, what we call this Groth's mechanism. Proton hooping means 1 H plus they are being donated to next water molecules. Then this next H plus is donated to next water molecule. Then these things keep going on. So the water accepts proton and becomes the hydronium ion. At the end, you can see they accept the proton, and they becomes the hydronium ion. So this is what your proton hooping fungi is. And definitely they ask that the basic question they ask, like water molecule exists in which form, then you have to say that it's a hydronium bond in water. And they can ask the question like why water is universal solvent, and they will give you 4 or 5 like mind storming option, and you will get confused कि किसको मैं टिक करूँ, ये तो ये भी सही लग रहा है, ये भी सही लग रहा है, ये भी सही लग रहा है. But my friends, I'm telling you, once you read thoroughly to these books, then definitely you won't face such kind of dilemma, and you won't face any kind of these dots in your mind. So like we all know that water have like slight positive and slight negative electric charge. So once you put some molecules or some salt or some things in water, so they dissolve, and they positive charge they go to their negative electric charge, like oxygen O2 वाला जो वहाँ पर negative del है generate हुआ है. And that's what I said, कि this H2O molecules, they carry slight positive electric charge, while the oxygen side carries slight negative charge. So when you dissolve the water, a few of the salt or few of the molecules, they dissociate and they move to their respective charge according to their overall surface charge. Next slide, please. So these are the questions they are being asked. So I have given some another example also, like in 2019, I have seen that there was one question in part C. Part C or part A, they have asked that why most of the non-polar amino acids, they are found inside the core. Actually, it's a wrong statement over here. Actually, your non-polar amino acids, they actually present inside the core, and your polar amino acids, they actually found at the surface. So this was the question they have asked. So why it is so? Why it is so? Then they have given 4 to 5, mindstorming like options, and then you have to choose the right one. So it's मैं हमेशा स्टूडेंट को बोलता हूँ कभी भी पढ़ाई को ना वो टैबू नहीं मानना है. सब कुछ आपके सराउंडिंग में ही है. You can just correlate the things with your surroundings. Everything is there in your surroundings. You will find each and everything in your surrounding. I will tell you how and why you correlate with these surroundings. So polar and non-polar, ये basic funda है. We all know, means our जो भी हमारे proteins हैं वो कहाँ रहेंगे. Let's say for example, if they are in the cytoplasm, let's say transcription हुआ, translation हुआ. After transcription, your messenger RNA, they are being exported to your cytoplasm. Now, after all kind of modification and everything, they have converted. After translation, they have converted into final naive, or finally, you can say not naive, you can say finally your native protein. So in that way, so why these non-polar amino acids, they are buried inside, and why these polar amino acids, they are covering and they are present all over, they are found at the surface of the protein. Why it is so? Because your cytoplasm, they are rich with 70%, they are filled with the water. So of course, same dissolve the same. So this is the obvious thing, that polar amino acid that should be present on the on the surface of the some of the globular protein, so that they can interact with the water, and this will finally help in they will keep this tertiary structure of protein intact or maintained. So these are the few questions that are being asked. Like also, in part A, the question that could be asked, like protein conformation with the lowest free energy. Like they will give you many options. Like then you have to consider, like the protein folding. You can see on the left side of the screen. This is the linear chain of polypeptide, and this last one, the lower one, is your native polypeptide. Native poly to tertiary structure, or folded or completely functional protein. You can see on the surface. The maroon, you can see on the surface in the orange color, dark orange color, you can see. So all of the hydrophilic or polar amino acid, they just occupy this periphery or surface of this protein, so that they can interact with the water. Okay? And this blue wala, that core, so this blue region is all of your hydrophobic or non-polar amino acids. They are actually occupied, or they have buried themselves in their core, so that they cannot, because they are hydrophobic, they don't want to interact with the water. So because of that reason, they try to bury themselves in the core of this structure, so that they can minimize the interaction with the water, or something like that. And this structure First one is unfolded and last one is native. So I'm just providing the glimpse over it. So I'm not taking some class, don't take it otherwise. So it's just a glimpse I'm providing which kind of question means this CSIR they usually they ask. So they will ask, like they will give you the two structure. One is unfolded protein, another one is native protein. They will ask which one has the lowest free energy. Then definitely we will have to say this native one, this protein conformation with this native structure, they have like lowest free energy. मैं इसको भी आप लोगों को coming slide में आपको एक चीज आप लोगों को बताऊंगा protein folding. Then I will let you know about this journey of this protein folding also, from which this question are being asked by the CSIR and other funding agency. So these are the questions they are being asked, and you know one of the like जो CSIR का जो एक अच्छा और गेट वगैरह का और DBT का जो पेटेंट काफी ये फेवरेट क्वेश्चन होता है, लाइक आपके जो once your protein is getting folded, so which kind of bond is predominating? It's a covalent or non-covalent? Then of course it's a non-covalent. A non-covalent maybe they will give you many options like hydrophobic interactions, hydrogen bonds, like ionic bonds, van der Waals interactions. Then which interaction is they predominate during the course of folding of protein. So these are the hydrophobic interaction. These hydrophobic interactions, they play crucial role in attaining the tertiary structure or native structure of the protein. They can ask you, in part A, they can ask you the question from enthalpy, entropy, that whatever you have studied during your +2 and your graduation classes. So they will ask all kind of basic fundas from enthalpy, entropies, and these things. Next slide, please. Rajiv sir, next slide. Yeah, so they will also ask, these are the few questions they also ask, why I'm emphasizing this biochemistry and molecular biology, this biochemistry and molecular biology. 50% of the question, these are being, covered by your biochemistry and molecular biology. Biochemistry, molecular biology, they cover almost more than 50%. You can say 50% part they are covered by biochemistry and molecular biology. So we can see, these are the simple polypeptide chain, and we all know that there is a, you can see peptide bond, and we know that peptide bond, they are rigid in nature. They are partial, double bond in nature, and that's why they are the rigid in nature. So there was a question in part C. They have asked that if peptide bond is rigid in nature, then how come your protein attain the range of 3D conformation? How is it possible if your peptide bond, because one amino acid they are attached to another amino acid through peptide bond, then how come they are so flexible from the linear polypeptide chain, they fold and they finally convert the native or tertiary structure? So how it is possible? So then here the concept of phi and psi bonds comes into the picture. These are actually your C alpha and C, what we call psi bond, and C alpha and what we call phi bond. So these phi and psi bond, they play a crucial role during your protein folding, and they provide more and more flexibility to your polypeptide chain to attain the tertiary structure. Next slide, please. And they will ask propensity of, they can ask the question from this region also, like proline, glycine. The proline and glycine, you will always find one question from the proline things, like या तो वो पूछ सकते हैं कि what is the uniqueness about this proline amino acids. So you will find always one question from this proline and glycine, because these two amino acids, they are very important to attain because they are on this surface and all wherever turns are there in the protein structure, you will find this glycine and prolines over there, because this proline they helps in inducing the ning, and glycine because they have R group as only H, so they are more flexible. So that's why they are present on the turn in 3D structure of your protein. So the usual question they ask, why proline and glycine is known as helix breaker? Means helix breaker, why means आपके जो भी proline है या जो भी glycine है, why it creates instability to the helix, to the alpha helix? Why it is so? So you know the answer, because you have studied the Leninger, you have studied the basic things about these amino acids. Because this proline, they have like rigid ring. They have you can see they have imino ring. They have very rigid N-C alpha bond. Because of that reason they will produce ning in the You can see in the figure, blue color, that right side one, blue color right side one, this bent, this link is over here. So it's because of proline. So, you know, that's the reason your proline, they are not found in the alpha helix. And your glycine, they are two, you know, या तो जैसे हम लोग ये बोलते हैं ज्यादा स्ट्रिक्ट भी नहीं होनी चाहिए और ज्यादा फ्लेक्सिबल भी नहीं होनी चाहिए, you know. So similarly, your glycine, they are too flexible, and your proline, they are too, you know, they are too tight. तो ऐसे भी नहीं होनी चाहिए. That's why these proline and glycine, they have been excluded from the alpha helix during the course of evolution, you can say. So these are the reasons. Next slide, please. Then of course you will find a few of the questions in alternative year, like if they have missed this year, then definitely they are going to ask the question from this Ramachandran plot. So this Ramachandran plot is, again, it's a phi or psi versus, and this is, you know, this sterically allowed conformation. In this plot you can see all those sterically allowed conformation, because, you know, first of all, there is a polypeptide one, sorry, peptide one over there, so they are the rigid, and, you know, one amino acid, they are connected through another amino acid through this peptide bond, and this bond is rigid, but there is another third amino acid and fourth amino acid, they are connected through, you know, they have like phi and psi bond, but still, you know, because once they fold, so their R group, they try to come closer. So, you know, they might create some kind of steric hindrance, they might repel, and they might not allow the protein to attain the particular structure. So this is what we call Ramachandran plot. The all kind of sterically allowed conformation, they are beautifully well explained in this Ramachandran plot. You should read this plot, and definitely one or two question is going to ask from this from this topic as well. You know, they will give you the 4 box, this 4 quadrant, and, you know, they will ask, they will write A, B, C, D in 4 quadrant, and they will ask, what is A? You have to say it's a sheet. Then what is B? Then you have to say it's a helix, or something like that. The position of right-handed alpha helix, position of alpha-handed alpha helix, left-handed alpha helix, position of right twisted beta sheets, position of collagen, position of other things. Next slide, please. Then of course they are going to ask this thing, like this question has been, I have directly taken the snap from the CSR exam, not from CSIR, actually this was asked in GATE exam. So this, you know, this position of glycine and proline in the Ramachandran plot. As I said, this proline, because they have R group, only H as a R group, so they are super flexible. Glycine is super flexible, so, you know, they can attain any of the coordinate. But, you know, proline, they have, you know, they have rigid N or C alpha. They are not flexible, they are too tight. So they occupy in particular this quadrant. So something like that. Next slide, please. They will give you the box. They will give some A, B, C, D, or then they will ask, what is this? You know, I was saying ki protein folding. So protein folding, it's again, it's a journey. It's a journey. Like हमारी journey होती है. We start, हम लोग schooling करते हैं, then we go for +2, then we go graduation, then we go for PG, then we go for PhD, higher study, blah blah, something like. So PhD also, sorry. So protein, they have also their journey, protein folding. So, you know, once first they are linear polypeptide, they attain some local conformation of what we call secondary structure, alpha helix or beta sheets. Then further they, you know, keep on folding, and finally they attain the tertiary structure, something like that. You must have seen that Transformer movie. I used to say, you know, things, you will always find the concept around you. You must have seen this Transformer movie. You know, in Transformer movie, अगर आप लोगों को याद हो, different parts होते थे, वो एक साथ once they comes together, और वो complete एक huge structure में convert हो जाता है. Then that structure that robot, they start fighting, something like that. So similarly, you see this protein folding. What we call, you know, we call them this molten globule. This molten globule has been also directly asked, and you will always find some question from this region also, from this part also, this protein folding, what we call them hydrophobic collapse, we call them molten globules, okay? And we call them energy funnel also. You can see they looks like funnel shape. So that's why we call them energy funnel. So initially at this stage, this entropy is very high, because protein, your polypeptide is in linear form. They keep on folding, they attain the tertiary structure, secondary structure, then tertiary structure, then further fold, and they finally attain, you know, their native structure, what we call 3D structure or functional structure. And, you know, and at that region, at tertiary structure, that native structure, you know, that del G is overall negative. And that's the reason you can see over here, entropy is decreasing from top to bottom, okay? And free energy is decreasing, something like that. So this is what we call hydrophobic collapse. So these are the, again, this was the journey of protein folding, and questions are being asked from this portion also. Next slide, please. So then, of course, there will be a like protein denaturation and renaturation things. There was one famous experiment that Anfinson has given in 1950. 1950 में इन्होंने 1952 में इन्होंने दिया था ये experiment, Anfinson experiment. You know, they asked the question from this part also. So I will tell you one more thing. This is the complete Anfinson experiment. You all must be knowing about that. It's clearly well explained in any of the good book of biochemistry. You know, they have proved like how first they denature the protein, then they remove the denaturants, and then finally the protein got renatured, something like that. And you know, one question was directly asked in 2016 from this Anfinson experiment, and you will get surprised to know that, you know, because here in this picture they have said that there are 8 cysteine residues. So they had asked one question. They had given this cartoon. They asked in how many different ways 8 cysteine residues could recombine at random to form 4 disulfide bond. You know, so this is again it's from biochemistry, but you know, they will take you to probability, they will take you to permutation combination, they will take you to somewhere else. So they used to ask questions something like that. So you can answer this question like 7 into 5 into 3, something like one different combination. You can work out on these, and you will find this question in your this Leninger also. So that's what I said. You will get few of the question directly from, if you have studied biochemistry from the Leninger, then definitely you will get few of the question directly from this book itself. And you know then your melting temperature, TM and all. So how GC and how this like AT or GC content is directly related to your melting point of your. Sorry, it's all about no. I was discussing about DNA. So it's a like melting point, means once 50% of your protein gets denatured or something like that, means your hydrophobic and other kind of interaction, they are being disturbed by exposure after exposure of these denaturants and all. So you all have idea about this experiment as well. So you know the questions are being asked from these areas. I have highlighted over in the green. So you know they will not ask directly what is Anfinson experiment. Yeah, they can ask. There was a question that was they have asked that in this DBT exam. They have asked that like DBT JRF, like Anfinson has conducted this an experiment. So what was their conclusion? So they will give you 4 or 5 points. So in that way also you can answer this question. Next slide, please. So protein, then because biomolecules like protein, nucleic acid, lipid, carbohydrate, all things are there. So you know you have to read all those biomolecules. And basically, if we see protein, nucleic acid, they are basically majorly covered. And of course, somehow your this lipid as well. Lipid, they ask very few questions from biomolecules, lipid and carbohydrate, but major portion they ask from this protein and nucleic acid. That's why I have emphasized few of the sample question I have given. Like for the DNA, if you see, you all know about this hyperchromic effect. You know, once you attend some interview now, that's why I am telling you to read or prefer some good book. You know, when I was attending an interview of TIFR Mumbai, so they have asked that, Tushar, you have given with 2 test tube. One test tube is having like denatured DNA, one is having complete DNA. So how you will found which test tube is having which test tube is having separated strand and which of the test tube is having intact double-stranded DNA, something like that. So this is what here your hyperchromic shift or what we call hyperchromaticity effect. It comes into the picture because we know that in double-stranded DNA, your nitrogenous bases, they are buried in the stack or in the core because they are hydrophobic in nature. So and you know that DNA they show strong absorption at 260 and protein 280. So 260 they show the strong absorption. It means that, and these absorption they are because of their nitrogenous bases. So in double-stranded DNA, your nitrogenous bases they are buried inside. So they don't show that much absorbance. But once they are denatured, they convert into single-stranded DNA. So now their nitrogenous bases are exposed. Now they show the increased absorption of UV rays, something like that. So they will ask you the question like there is a free nucleotide, single-stranded RNA or DNA, then double-stranded DNA. So how you see the hyperchromaticity in this aspect. Next slide, please. Then coming to the next part, like cellular organization. So again, this A portion is membrane structure and function. You will again find this portion in lineager, chapter number 11, chapter number 12. 11 is there is signal transduction, but 12 you will find this in chapter number 12 of the lineager. And then B and C, and then again D, B, C, D you will find in this CARP. I have given the logo of the snap of this book also, Gerald CARP. Gerald CARP. It's a book for cell and molecular biology. So you have to see, now it's a cell biology. I said कि पहले biochemistry पढ़ो lineager. Now I'm asking you to read cell biology for this. And in these books, don't go like they are too thick. इतना मोटा book है भाई, कैसे मैं cover करूँगा या करूँगी? But there are so many portions, they are being overlapped. कुछ lineager में, कुछ CARP में, कुछ Watson में, you will find so many chapters. They are overlapping. So by smartly you can ignore those regions, and you have to play smart. Bit. So if we see this membrane structure and function, you will find this portion in your lineager. This chapter B and C, it's a completely all cell organelles like cell wall, nucleus, mitochondria, Golgi, lysozyme, peroxisome, endosome. They also ask questions from this region. And the protein sorting, like how your protein after we all know that in case of eukaryotes, this protein after transcription inside the nucleus, they gets all kind of modification like tailing, capping, and this removal of splicing things and all. Once this mature messenger RNA they enters into the cytoplasm, okay, now they have to choose the path. If proteins are destined for the cytoplasm, then they will follow the bare cytoplasm wala pathway, bare ribosome wala pathway. But if this protein, the fate of this protein is for for endomembrane system, like for ER, Golgi, lysozyme, then they have to follow the endomembrane wala pathway. Here your signal hypothesis and everything comes into the picture. So this chapter is also very important for this CSIR, GATE, DVT, JRF point of view. Then there is like microbial physiology. So microbial physiology, there is a book Peljar. You will find very concise things are there, like for bacteria, for viruses, for everything they are there. They will always ask, from the RNA world hypothesis. You will always find few questions from RNA world hypothesis. What is RNA world hypothesis? Why it was, you will always find one question like why like DNA has thymine at the place of uric acid. Why it is so? And you will get all the answer of this thing once you read this lineager, like Watson, this Gerald CARP. You will face all, definitely you will face one of these questions while studying these texts in the details. Next slide, please. Yeah, so just I have given one simple example for this biomembrane or biochemistry, plasma membrane region. There was one question in 2011 or 12 they have asked, like why plasma membrane of polar bear has high amount of unsaturation. You all know that there are two types of fatty acid, like saturated and unsaturated one. So unsaturated has, we all know that. You all know that. Unsaturated has double bond, and this saturated has a single bond. So if you can see on this pictorial representation over here on the screen, the saturated acid, fatty acid, they are compact, they are closely packed and compact. But if you see this unsaturated fatty acids, because of this double bond, they some sort of link is over there. So they are not closely packed. So this is the region. This is one of the reason. Because what is the nature of this plasma membrane? It should be fluidity, and so that some of the molecules they can come from outside of the cell to inside the cell. They can travel through the plasma membrane. Everything they travel from the plasma membrane. If your membrane is too much rigid, if they are too rigid, then it's not possible to all kind of trafficking, molecular trafficking will be completely shut down. So for that reason, unsaturation should be there. So this is the reason, this plasma membrane of polar bear, they have very high amount of unsaturated fatty acid. This keeps their plasma membrane fluid in nature. So this is the nature of plasma membrane, so that they can take the things from the outside the cell through endocytosis, and they can release the things from outside the cell through the exocytosis, something like that. Next slide, please. Yeah, from the cell biology, I was saying that signal hypothesis. These are the questions they will ask. They will ask. These are the most patented question by CSIR, DVT, GATE. They definitely they ask if they have not asked this year, they will ask in next year. So something like that. These are the whatever I'm giving the sample over here. These are the these are somehow they have been asked somewhere and they will be asked in coming future also, in coming time also. So you have to prepare in that way. You have to prepare so that you can you won't face any problem in examination hall. Like this signal hypothesis, as I was saying, that once you're in eukaryotic cell, once your mature messenger RNA they comes out the nucleus, now they are ready for translation. So now if the fate of this protein is to the endomembrane system, बोले तो अगर आपके Golgi, lysozyme या plasma membrane से बाहर जाना है, then they will follow the endomembrane system. Then these protein must have to follow the signal hypothesis वाला फंडा. And जैसे आप लोग गेम भी देखते होगे, कुछ भी देखते होगे, there is always destiny for that particular protein. ऐसा नहीं है कि protein बन गया, now they are asking where shall I go. No, it's not like that. During translation time, at the end terminal of most of the polypeptide, they have signal sequence. This is what we call sorting sequence. If this signal sequence, if this says no, your final destination is in nucleus, then definitely protein will enter into the nucleus. If they have like that, if the protein is wearing red cap, means it has to go to the nucleus. If they are wearing green cap, they have to go to Golgi. If they are wearing purple cap, they have to go outside the plasma membrane. So this cap is nothing, it's a signal sequence. And signal sequence are nothing, they are in the form of amino acid. So the question could be asked from this region also. And the folding and further post-transcriptional modification inside the ER, you can see the this lower panel figure, you can see over here. So how these, 3 molecules of N-acetylglucosamine, then this glucose, 9 molecules of glucose, then mannose, they are being added in the ER, and what is the role of them, how they are being added. So these kind of questions they are being asked. Next slide, please. Yeah, then again protein trafficking through endomembrane system. As I said, once protein has entered into the ER, now they will move to Golgi. And if the destination of protein is once protein has entered ER, there will be a particular modification, then protein will go for Golgi. There will be again some sort of modification in the Golgi. And if the final fate of this protein is the ER, then protein has to again come back to the ER. So for and this kind of trafficking, they are mediated by COP1 and COP2. For every if you go to some traffic, you will always find some protein, some police standing over there. So these are the things you can correlate the things with your. So average, we are the human being. We should learn from ourselves. They are highly disciplined and they are very efficient. So we should learn from them. So these are the questions that are being asked. Next slide, please. Then, of course, if we talk about this molecular biology, this operon-related question they are asked, definitely they will ask few of the question from the lac operon, few of the question from, the strip operon and all. They will ask like full expression of lac operon requires lactose or cyclic AMP, allolactose or cyclic AMP. Then, people get confused between lactose and allolactose. So all things, you should study in minutely. Then, so how you will study these things in minutely? For that, you have to have, you have to have some good textbook and you have to refer those textbook as well. Then definitely like people can ask if there is a deletion mutation in operator of lac operon, the expression of lac gene will be permanently stopped, constitutively expressed, not expressed, resistant to catabolic expression. What could be that? Then you should study the you should have idea about how if there will be mutation in operator, then it means that repressor protein that won't be able to bind to the operator. It means that it will be constitutively expressed, something like that. So they are the very basic question they ask, and very basic and conceptual wise, very I would say they are very they ask very conceptual and analytical based question. Next slide, please. Yeah, then they ask the cell cycle checkpoints also, like CDK, cyclines, and role of cyclines and CDKs. Like जैसे मैं आप लोगों को बताता, once you right now I'm in the Bhagalpur. If I go from Bhagalpur to Patna, I will find so many things. I need to cross so many districts in before I reach the Patna. So at each district, there will be a banner over there. So this Munger police आपका स्वागत करती है, something like that. So you will find always post something like that. So this happens with our cell division also, cell cycle also. If there is a G1 as G2, so at the end of the G1, then definitely one cyclin will come into the picture. They will say, dude, now you have to stop everything. Now you have to prepare for S phase, because S phase is for DNA synthesis. So you have to, you have to make the things ready for the S phase, because you are going to need lot amount of protein, lot amount of polymerase and other proteins to pursue the DNA replication. So keep yourself ready for that thing. So these cyclins and CDK, they keep ourselves ready. And these cyclin and CDK, most of the time they act as a transcription factor. They either they express or they either they express or they down regulate your expression of some sort of gene. So you should study these things also in detail, and you will find, Gerald, Karp, and Albert. You will find these things are beautifully explained in those books. Next slide, please. Yeah, for this this fundamental process, it's a complete molecular biology. I have written over here 50% part is covered by biochemistry and molecular. अभी तक जो मैंने आप लोगों को बताया 1, 2, and 3. These three regions, they covers the 50% part of your this CSR, DBT, and things like that. Here you will find all of the like DNA replication, repair, recombination. They will ask like, why promoter are highly AT rich? Why your origin of replication they are rich in they are highly rich in AT or something like that? Why it is so? So they will ask the question something like that. So you should act accordingly. You should act in a smart way. And they will ask like, what is processivity, what is fidelity, and they can ask some Michelson style experiment from here, a very famous experiment that have proved that DNA is semi-conservative. So Michelson style experiment. So there are 3 experiments. They have nicely explained. Means DNA is not a conservative. It's not a disperse. It's a complete. You can see it's a semi-conservative. And there is experiment Michelson and Starr they have explained. So you should study those experiments in detail, and you should keep there like how they have done this experiment. You don't have to mug those things. You don't have to कुछ लोग नहीं कुछ होता तो let's mug it. इसको रट लेते हैं. No, don't mug anything. There is nothing to mug. You will always find some concept behind it. Try to get that concept. Try to knock to that particular concept. You will get it, and you will never. Trust me, my friend. You will never forget about that. If you once you understand that particular concept. So like we have like all kind of like they will ask, question could be asked from all kind of DNA damage, repair, like transcription, translation, gene silencing, RNA editing, and like post-transcriptional modification, like transfer RNA. If you see in case of eukaryotic transfer RNA, there is CCA at the 3' end. So this CCA, one question was asked in GATE into because in my student, so this time also they have 4 out of 5 students, 4 students they have cracked the CSIR this time, and one student C got placed in ISC for PhD. One of our student, her name is Prachi, so she just got placed to the ISC Bangalore. So this question, she said that sir, this question was directly asked in that question. That transfer RNA. I used to teach them during post-transcription modification. There is a CCA at the 3' end. So this CCA is not the part of gene. Actually, this part is being added after the post-transcriptional modification. So these are the minute things. These are small things. And my friends, trust me, you will always find this answer in your Leninger, Watson, and all these books. Trust me. So just blindly follow this book if you really want to pursue your career in this area or higher.
Tushar, your voice is not audible to us.
Hello. Am I audible?
I guess it's a network issue or something. It's breaking, in fact.
Hello, sir.
Yeah, Tushar. Now I think you're audible to us.
Yeah. Please. Yeah. So next, yeah. So whatever I said, so these three regions, they are like they are majority of questions they are being asked from these three. Next slide, please, sir. So yeah, this is the cell. Fourth one is cell communication and these things. So this is again, it's a complete, it's a package of cell biology and immunology. You can see over here in B, cell signaling. So there is chapter number 11 in the lineage. You will find this cell signaling. There they have beautifully explained all those questions. Okay. Then if we see in this first one, it's a complete immunology one. This cancer one, you will find this thing in your Gerald Carr book. And this E1 is a complete immunology. Immunology, they will ask like funda of epitope, paratope, what is continuous epitope, what is discontinuous epitope. And they will ask the question, some experimental aspect in part C, like they will ask the experiment of Portal and Edelman, how antibody has been proved that they are Y-shaped and they are heterotetramer, how it has been proved. So, these things has been explained in a nice manner by the Portal and Edelman, by these two scientists, and these are pretty well explained in your immunology books and all. And they can ask like antibody diversity, VDJ recombination. And I still remember in part A, once they have asked the question, I told you, they can ask you in part A, they can ask you anything. Physics, chemistry, biology, maths, non-maths, general knowledge, they can ask anything. Once they have asked like, why antibodies is always Y-shape in nature? Why nature has selected this Y-shape nature of antibody? Why not T-shape? Why not S-shape? Why not R-shape? Why always Y-shape? Why it is so? And they will give you so many explanation for that, and you have to go for best one. So these are the basic things. So you don't have to forget this base, and you have to build the story up to that level. So these are the things you have to keep in the mind. So then, of course, you have to go through this T cell and B cell activations wala things, and you will find this thing in immunology. Next slide, sir, please. Like that signal transduction that I was saying, directly they have asked so many times question like GPCR, what is the typical characteristic of G protein coupled receptor? Are they 7 membrane helix? So this is, yes, this is the typical, characteristic of G protein coupled receptor. They are 7 membrane helix. They are actually your membrane protein. You can see the green color one, and they are made up of 7 transmembrane helix, something like that. Then they will ask the question from G protein. So what are G protein? G alpha, beta, and gamma. Which one is functional one? G alpha or G? So what happens once GDP interacts with the alpha region, alpha polypeptide? What happens? Does beta and gamma leaves? And if it leaves the complex, what will be the next events? And what are these effector molecules like adenyl cyclase? How these cyclic AMP are being produced? What are those secondary messenger? And most important thing, they will ask like, why do we need this second messenger? Like, why do we need this transporter kind of things? Something like that. Because, in our body, if we see, we have like 2 hormones, like hormones which are proteinous in nature, and some hormones, they are steroid in nature. So hormones, those are steroid in nature, means they are hydrophobic in nature. So for them, there is no need of, transporter. They can easily cross your plasma membrane. But the hormones, those hormones which are proteinous in nature, they need some sort of transporter. So your insulin and glucagon, once you had your meal, आपने just खाना खाया, so तुरंत खाना खाया, you got a lot of this carbohydrate or this glucose. So this insulin will be activated. Once you are in fasting condition, means fasting, if you are on fast, then your this glucagon, they will be activated, and they will start churning out this glucose from the stored this glycogen. So something like that. This kind of question are usually asked in these exams. Next slide, please. Then these are actually your genetic engineering, what we call recombinant DNA methods. So the 10%, this region, they cover 10% of overall syllabus. In DBT, if you go DBT or GATE, they are majorly covered, this region. This method in biology, in GATE exam and DBT, they actually basically cover 50%. But in CSIR, they cover 10 to 15% this portion. So collectively, we see the biochemistry, cell biology, molecular biology, and this RDT. And I will tell you the last one, the techniques. These complete things, if you have studied all 4 or 5 things in a detailed way, no one is going to stop you for cracking these exams. So here you can see, I don't want to take much time because time is, I guess it won't allow me for longer period. So I will just summarize. I will just go a bit fast in this for these slides. So here you can, if you have any other queries and all, you can contact me anytime. So I'm ready to help you, or you can contact to Sanjeev sir for these things and all. So this is, this is your method in biology. It's your this portion is basically your this recombinant technology, what we call genetic engineering technology. They will ask the question like, what is alkaline lysis method for plasmid isolation? Means you don't have to study like you have to study each and everything in detail. Like if you are adding some chemicals, then you should know why we are adding chemicals. If you are adding glucose, if you are adding some sort of SDS in particular solution, then why you are adding SDS? What is actually happening at the level of cell? You should this should, while reading the book, this thing it should come in your mind. While DNA purification, why we add ethanol? Why we add isopropanol? What is the need of that? Why we do centrifugation in that step? Why we store DNA in T buffer or nuclease free water? These kind of questions, they are usually asked. Like so here also, like cloning things, like basic concept like restriction, digestion, like vectors. All you should know about all those fundas of vectors, like cloning of genes, then restriction enzymes, then complete ligation, then transformation, then recombinant protein expression, then purification. Whole thing you have to, and you will find these things in details in these 2 books, like T.A. Brown and some of the techniques book in Wilson and Walker. And then of course DNA sequencing, you have to also go through this DNA sequencing and all. Next slide, please. One question they have asked, like, while doing the gel electrophoresis, you see more bubble at cathode region, cathode electrode, and you feel less water bubble or less air bubble at anode. Why it is so? So these are the things, in part C they will ask, like let's say Mr. Pius, he was performing some experiment and he was loading the gel. He turned on the agar gel electrophoresis, and then suddenly he observed that there is more air bubble at cathode region and there is less bubble at anode region. So could you help him to explain the reason why it is happening? So something like that they can ask. Next slide, please. So like again, agar gel SDS page, they will ask why you are. I know that you all are knowing like SDS page, why we use SDS, what is the role of SDS, how it denatures the DNA, and what are the basic funda behind gel electrophoresis, SDS page, 2D gel electrophoresis. You should know all those things in the detail. All kind of techniques you should study in detail, because techniques is also a major question. Because once you go for once we go for part C, these techniques will help you a lot, because majority of questions they are being asked from this region, from this techniques wala region and all. So they can ask about like this isotachophoresis and what are the role of this, because where we have like 2 different sets of pH for stacking and separating gel. So you should so you should know all those things in detail, and you will get everything in Wilson and Walker books. Next slide, please, sir. Yeah, so question could be asked like, from these region also, like, as this is what I was saying, they will not ask direct question from these techniques, and also, and they will give you like one agar gel image. They will show there are 3 bands. So which bands is your, this is one of the favorite questions they are being asked in GATE. They usually ask. Means you have same base pair or same size of linear DNA, supercoiled and relaxed DNA, what we call nicked DNA. And if you run these 3 DNA on the agar gel, which will move forward and why? Something like that. So you should be able to answer it. And some SDS page and native page. In SDS page, we use SDS. Means we separate the DNA in denaturing medium. But in native page, we do not use SDS. We just separate the DNA in non-denaturing condition. So they will give you the 2-gel profile, and they will see that in HDS page, that same protein is showing of size of 100 base pair, but in native page, they are showing of 60 base pair. So why it is happening? Why it is so? So you have, you have to explain all those things in the detail. So why in native page they are showing of 100 kDa, and in SDS page they are showing of 50 kDa. It means that they are dimer. So something like that. You have to, you have to act accordingly. So questions are being asked from this, and they can ask, yeah, something like that. Next slide, please. So these are the sample I have given already here. You can ask the slides from Piyush sir and Rajiv sir. They can share the question, because these are the questions they have been asked in CSI or GATE or something like that. Next, they will ask from question from blue white screening. What are those? What is LECZ gene? What is alpha complementation? And they will ask a few questions from your plasmid, something like that. Next slide, please, sir. Like you can see over here, there is one question. Like a biochemist is attempting to separate a DNA binding protein, something like that. The clear-cut question is written over there. There is a table. Isoelectric point is given, size is given, and their property is given. Now there are 4 options, and out of these 2, which 2 3 are correct? You have to go for this. So in part C, you will get question like this. Exactly, you will get question like this. Here, question 1 and question 2 are given. So in part C, you will get the question like this. Because as I said, it's not a subjective, but you have to do the calculation, you have to apply your concept, you have to apply your this common sense, and then you have to find. So it will take some time, 4 minutes to 3 minutes, 4 minutes, it will take time, but you will be able to do it. Once you have, because all these things, see, you can see over here, they have asked question from agarose gel electrophoresis, they have asked question from SDS page, they have asked question from this affinity chromatography, size exclusion chromatography, or ion exchange chromatography. See, all 5, 6 things they have combined in a single question, something like that. So you should approach in that way. Then they are going to ask on the basis of PI difference, like 2D gel. Next slide, please, sir. Yeah, these are the questions. Next slide, sir. Part C, they will cover such question and something. So now, this is again, it's a technique portion. So like detect like ELISA, like different types of ELISA, like indirect, direct sandwich, RIA, Western blotting, Southern blotting. So these are the techniques. You will find this thing in Wilson and Walker. But I will tell you one thing over here. Wilson and Walker, you will get the things in much more detailed way. But there are some techniques like NMR. Like X-ray diffraction, fluorescence, like confocal microscope, some SPR, surface plasma resonance, AFM, atomic force microscopy. So these things, there is a book for this Pathfinder. You will also get one book, Biophysics and Techniques for Biophysics and Molecular Biology. So they have all these techniques described in, very crisp and very detailed. They are also in the depth, but in a crisp manner. So you can refer this book for these techniques. Next slide, sir. And they will not ask directly question what is sandwich ELISA and what is indirect ELISA. They will give you the some case study like that Dr. Pawan is doing so and so and so, and he observed, he got the results, something like this. So what could be the simple explanation for that thing? Again, these are the techniques like microscopic techniques, electrophoretic techniques. Next slide, please, sir. Yeah, it's a I will tell you one thing. It's a developmental biology. People basically get confused कि developmental biology में कहाँ से book मिलेगा, कैसे पढ़ना है. If you want to get the, if you want to, of course, we should study all things in detail. But here is the trick. I will tell you one thing, because you will see few of the things like A and B. You will get these things in in your plus two biology syllabus. You will get these 2 things. But this C wala portion, morphogenesis and organogenesis, you will find in chapter number 28, lineage. So you will find like they have considered example of Drosophila,
how this backward and nano gene, they are responsible for anterior and posterior regions. How these, these hunchback and cadwal genes, they are responsible for segmentation of Drosophila. So these are few of the questions they are being covered from this developmental biology. So I've highlighted this. You can focus on this this highlighted region, and you will get this thing in, this Leninger chapter number 28 you will get. And for other things, you can refer, there is a book, Gilbert, Developmental Biology. But I would say you don't have to stress too much on this developmental biology wala things. You just read chapter number 28, Leninger, and few of your +2 wala biology wala part. It will be cover most of the things. Next, sir. Next slide, sir. Then finally, that was all about, that was all about your higher studies, like biochemistry, molecular biology, cell biology, immunology, genetic engineering, and techniques, all sort of techniques and all. That's what we have covered so far. And if you have whatever I have said, if you just cover those things only, you will be definitely true. But you have to don't have to underestimate other portion also. But still I would say इस पर ज़्यादा focus इस region पर नहीं करना है. For plant physiology, animal physiology, classical genetics, ecology, and evolution. Why? I'm not saying कि completely ignore it. No, I'm not saying that. Means you must have studied during your 11th and 12th. This NCERT book must be along with you all for this physics, chemistry, and biology. You must have all these NCERT books. You just refer those books, and and you must have, all of you must have studied. I guess you all must have prepared for this medical and this engineering examination and all. So you must be having notes for these things for physics, chemistry, and biology. If you have some study, if you have taken some tuition or some tutor for this things and all. So for these things, they will never ask the question up to the level of that. There will be a few questions of master's and graduation level, but you don't have to worry for that. So you just go through your this NCERT level, NCERT based syllabus, whatever you have: plant physiology, animal physiology, classical genetics, ecology, and evolution. No need to give extra time on physics, because basics and medical level you have already prepared well enough during your medical preparation and all. Next slide, please. Yeah. So now coming to the very last one, like part A. We have covered part B, part C. Part A. As I said, they will ask general science. They will ask quantitative reasoning analysis. They will ask the research aptitude. And I have given the snap of one book, R.C. Agrawal Mathematics. You just refer probability, permutation, combination things from this book. And directly, I will tell you, in CSI or GATE or DBT, not in the DBT, in CSIR and GATE, you will get few questions directly from this R.C. Agrawal. From if they are going to ask from probability or permutation combination. There are so many things in that book, but just refer probability, permutation, and combination, and somehow set if you have enough time. So and they ask very basic question. They ask general knowledge. Sometimes they will ask general knowledge. Last year in 2020 only, they have asked that, like they have asked like PM Fellowship Award is basically given for which institution? Central University, State University, IITs, ISC, NITs. Then of course you have to say it's for IIT and ISC. So you have to be update with your this general knowledge also little bit. If you see the news channel and this thing, but then I think it's more than but not follow the news channel of these days. They are bombarding with the Ria and Sushant Singh case. And yeah, so the basic and fundamental biology and physics to check the research aptitude. They will ask basic question from chemistry, physics. Next slide, please. Like physics, they will ask next slide, please. In physics, they can ask question like, how do birds sit on high voltage power line without getting electrocuted? उनको कोई करंट नहीं लगता है बैठे, क्या रीजन हो सकता है? We all have studied during our +2 in physics and all. So see the level of exam. So like they will ask, in how many ways can 6 ring of different types be had in 4 fingers? In how many ways 4 letter be posted in 3 letter box? In how many ways 8 student can be arranged in line or a circle? This question third, how many ways 8 student can be arranged in a line or circle? This was directly asked in part A last year. So so these are the few, these are the sample questions you can follow and you can go for it. So they just want to see your common sense, and they want to see your general science things and all. Next slide, please. Yeah, thank you. This is all I have. So because I have considered CSIR as a prototype, like I was more focused for CSR, but by covering these portion, whatever we have discussed so far, this will help you in cracking GATE exam, TVT exam, ICMR exam as well. So once you cover once you cover all the syllabus, whatever we have discussed so far, so it will be helpful in cracking all those exams. Thank you, sir. Sanjeev sir.
Yeah, thank you, Tushar. Or just to remind all the old days for us. बिल्कुल वो पुरानी यादें और पुराने क्लास आपने याद दिला दिया मुझे, तो लगा जस्ट मैं ही क्लास ले रहा हूँ सारे टॉपिक्स, और एज इट इज मतलब जो पोर्शंस में आप लोगों को हाईलाइट करता था, एज इट इज जस्ट यू हैव डन। ये बहुत अच्छी चीज है कि अच्छी चीजों को फॉलो करना। एक चीज मैं क्वेश्चन ऐड करना चाहूंगा, इसमें थोड़ा सा छूट रहा है उस चीज का, जो आप लोगों को मैं एक चीज लेता था कि स्ट्रैटेजी, यू हैव नॉट डिस्कस अबाउट द स्ट्रैटेजी। मतलब अगर बच्चे, जैसे ये तो आपने पूरा सिलेबस की बात किया, और आप लोग को मैं बताता था कि आप अगर आपका मैथ्स ये वीक है तो आप बायोलॉजी से कैसे बाहर निकल आओगे, या आपका केमिस्ट्री है तो कैसे आप निकल आओगे। ऐसे कुछ स्ट्रैटेजी लोग पूछ रहे हैं कि व्हाट आर द मिनिमम कैपेबिलिटी वी शुड टू डू टू क्रॉस द एग्जाम। मतलब क्या उसका मिनिमम जो सिलेक्शन पॉइंट है वहां तक पहुंचने के लिए कुछ एक स्ट्रेटजी प्लान आप लोग सजेस्ट करते हैं अभी ये में?
Sir, I will tell them to sir, just to cover those all higher studies portion for cracking the CSIR GATE and things like cell biology, molecular biology, immunology, genetic engineering, all kind of techniques and basics, and apart from that basic +2 level of chemistry, biology and physics. This is more than enough. Physics I don't।
ये कट ऑफ लेवल क्या रहता है उसको थोड़ा बता देंगे तो लोगों को प्लान करने में आसान होगा कि कितना वो मिनिमम तैयारी करके जाएं। जैसे हम लोग आप लोग को जेनी और जैम में कट ऑफ देते थे, तो एक प्लान बना लेते थे आप लोग कि सर ऐसे ऐसे ये पोर्शन कवर करेंगे तो हम लोग उसको अचीव कर सकते हैं। तो वो कट ऑफ लेवल अगर थोड़ा सा डिस्कस कर देते आज के डेट में, है ना। हेलो, क्या वो बाहर तो साथ डिस्कनेक्ट हो गए क्या राजीव?
डिस्कनेक्ट हो गए हो।
अच्छा, तो मैं थोड़ा सा तब तक कंक्लूड कर दें कुछ चीज इसमें। ये बात जो आप पार्ट ए का डिस्कस कर रहे थे, इवन आपको लाइफ में भी कई जगह पे ये होगा कि जनरल साइंस पढ़ने का हायर स्टडीज में आपको टाइम एक्स्ट्रा नहीं मिलेगा। यू विल नॉट गेट द एक्स्ट्रा टाइम टू कवर ऑल दोस थिंग्स। तो इसके लिए हम लोग बहुत अच्छा आप लोग का प्लान बनाते थे। हम लोग ने भी अपना ग्रेजुएशन में इस चीज स्ट्रैटेजी को फॉलो किया और मास्टर्ड में भी फॉलो किया। इसलिए मैं सजेस्ट ये करूंगा उसके लिए कि, और बच्चों को भी मैं सजेस्ट करता था कि आप लोग ग्रुप डिस्कशन किया कीजिए इस चीज पे। मतलब डिसाइड कर लीजिए कि मैं आज जनरल साइंस से बात करूंगा, आज मैथ से रिलेटेड अपना बेसिक बात करूंगा। मतलब कोई टॉपिक्स आप रख लीजिए और उस पर ग्रुप डिस्कशन कीजिए। डेली नहीं तो कम से कम अगर वीकेंड पे भी सैटरडे संडे एटलीस्ट संडे को भी मिलते हैं आप अपने ग्रुप के लोग और उसमें बेसिक्स से डिस्कस करते हैं, तो ऑलमोस्ट वो सब रिमाइंड करने का चीज होता है। और हर बंदा कोई ना कोई पोर्शन में स्टॉक, किसी का मैथ्स अच्छा होता है, किसी का केमिस्ट्री अच्छा होता है, किसी का जीके अच्छा होता है, कोई न्यूज सुना रहता है, कोई पार्टिकुलर पेपर पढ़ा हुआ रहता है। तो ये सब चीजों से क्या होता है कि आपस में शेयरिंग करने से बेसिक के लिए आपको पढ़ने की जरूरत नहीं पड़ेगी। बस आपस में डिस्कस करते रहेंगे। 3 ईयर इज सफिशिएंट। आप समझ जाइए कि एक साल में 52 सप्ताह होता है, और अगर 3 साल आप पढ़ रहे हैं तो 50 × 3 मतलब 150 क्लास आप डिस्कस कर रहे हैं, 150 क्लासेस। तो ये दैट इज द एनफ टू कवर ऑल द बेसिक थिंग्स एज वेल एज द इम्पोर्टेंट थिंग्स ऑल्सो। उसमें आपको शेयर कर सकते हैं। दोस्त के बीच में एक छोटी छोटी प्रॉब्लम होता है जो कभी कभी साई नेचर होने के कारण हम क्लास में डिस्कस नहीं कर पाते हैं, उसको आप डिस्कस कर सकते हैं। तुषार के बात से 3 4 5 बात निकल के आ रही थी, कॉमन बात कि ऑलवेज सेलेक्ट द बेस्ट बुक, ऑलवेज सेलेक्ट द बेस्ट सर्कल, ऑलवेज नो योर सेल्फ एंड एग्जीक्यूट योर सेल्फ पॉजिटिवली। इन सब चीजों को अगर आप ध्यान रखिएगा तो मुझे नहीं लगता है कि कोई भी ये सफर है जो आप पीछे रह जाएंगे। और बड़ा अच्छा लगा कि ये सब स्टूडेंट जो है फॉलो अप किए और आज इतने अच्छे जगह पे है। हमारा टारगेट था कि कम से कम 500 स्टूडेंट, मैंने जो अपना इंस्टीट्यूशन खोला था तो इस तरह का पेड़ एक अपना एक एक्सपेक्टेशन था कि ये संजीवनी ब्रिज कम से कम 500 ऐसे फल जरूर देगी समाज को कि जो बिल्कुल ये कहे प्लेटफॉर्म पे आके कि आई एम हेयर ड्यू टू संजीवनी। तो शायद मैं अभी तक वहां तक नहीं पहुंच पाया हूं, हमारी कोशिशें जारी है। these things also organized for all those covering that mission also कि these platforms are providing the benefit of the students and after getting the help or cooperation from this platform they will surely be successful। और क्वेश्चंस ले लीजिए राजीव, और किन्हीं का है तो थैंक यू। कोई है? संदीप सर का देखिए, क्वेश्चन ले लीजिए तब तक। संदीप सर भी है।
संदीप सर का एक्चुअली देयर फ्यू स्टूडेंट्स।
अच्छा, ये क्या तुषार रिकनेक्ट नहीं हो पा रहे हैं क्या?
हाँ सर, आ गया हूँ सर।
आ गए। अच्छा, हाँ ले लीजिए क्वेश्चन। अच्छा, कट ऑफ के लिए मैं बात कर रहा था, कट ऑफ थोड़ा बता देते, एक्सप्लेन कर देते। कट ऑफ
सर अगर पार्ट 1 में हम लोग देखें सर तो 15 क्वेश्चन अगर होते हैं, अगर हम लोग सर 10 क्वेश्चन को भी 10 से 11 क्वेश्चन को भी अगर हम लोग ये करते हैं, अगर हम लोग उसको ये करने की कोशिश करते हैं, अगर सर 10 क्वेश्चन भी हम लोग अगर कॉन्फिडेंट एनफ हो, 10 क्वेश्चन को भी अगर हम लोग टारगेट करें तो इट्स ओके, मतलब वो चीजें हो जाती है। और इस वाले में सर अगर 35 क्वेश्चन है सर, 35 में से मैं स्टिल बोलूंगा सर जस्ट पास करने के लिए सर 20 से 25 के बीच रहे तो ठीक है सर। और अगर उसमें हम लोग देखें 25 क्वेश्चन है, 25 में से 12 भी अगर सर सही बन जाए, 12, 13, 14, तो डेफिनेटली he or she will be through.
Yeah, I think someone is asking the minimum target portion, and I think she got the reply from this thing. Any more questions from the participants?
Sir, Mamta Chaube is there. She's asking, sir, just wanted to know from overall 13 units, how much units should be covered to clear the exam?
यही क्वेश्चंस को मैंने एक्सप्लेन किया अदर वे में। दिस थिंग यूनिट, चूंकि ये पर्सन टू पर्सन वेरी करेगा ना, आपको यूनिट डिसाइड नहीं कर सकते, या स्टूडेंट्स खुद डिसाइड करें कि उनके लिए कौन सा यूनिट है। ये कट ऑफ के पॉइंट ऑफ व्यू से, जैसा तुषार ने एक्सप्लेन किया कि आपको 12 यूनिट्स आपको करना है, 12 क्वेश्चन करना है, तो आपके पास पूरा सिलेबस है, आप देख लीजिए आप किसमें कंफर्टेबल हैं। और कोशिश ये करना चाहिए कि पार्ट बी और पार्ट सी में जो पार्ट कॉमन है उसको पहले अटेम्प्ट लें, ताकि आपका एक ही पोर्शन से दोनों पोर्शन कवर होगा। ये एक बहुत बड़ा स्ट्रैटेजी हम लोग बताते थे आप लोगों को भी।
यस सर।
और एनसीईआरटी से भी बहुत सारे कुछ क्वेश्चंस डेवलपमेंटल वगैरह से रहता है, जो बिल्कुल स्ट्रेट फॉरवर्ड रहता है, मतलब जूलॉजी और बॉटनी और ये इकोलॉजी पार्ट से बहुत ज्यादा वो ट्विस्ट नहीं करते हैं क्वेश्चंस को, तो आप वहां से भी 2-3 क्वेश्चंस को टारगेट कर सकते हैं।
वही सर मैं बोल रहा था, जैसे कुछ स्टूडेंट होते हैं वो परेशान होते हैं कि जो भी जूलॉजी या बॉटनी जो भी पार्ट है, एनिमल फिजियोलॉजी, प्लांट फिजियोलॉजी, कहीं ऐसा तो नहीं है इनके लिए भी हमें मास्टर्स या ग्रेजुएशन लेवल की पढ़ने की जरूरत पड़ेगी। मैं सीधे बोलता हूँ नो, अगर आपके पास एनसीईआरटी है या फिर अगर आपने मेडिकल का प्रिपरेशन करा है तो आपके पास डेफिनेटली वो नोट्स होंगे, दैट्स मोर देन एनफ।
जी। एनी मोर क्वेश्चंस फ्रॉम अदर पार्टिसिपेंट?
Manisha Kumar is here. Sir, you are an excellent mentor. She is complimenting you. Just one line for you. As a student, I feel you are professor to student. Thank you so much. An interest rising.
Thank
She
you.
is complimenting you.
ये इससे बड़ी खुशी तो और कुछ हो ही नहीं सकती, जो मैं अभी फील कर रहा हूँ कि हमारे स्टूडेंट जब सक्सेसफुल हो जाते हैं और वो आपको दक्षिणा के रूप में ये सब रिमार्क्स जब देते हैं, दीजिए और द रियल दक्षिणा, वो तो मैंने सर्वाइवल के लिए हम लोग पी लेते हैं थोड़ा सा, लेकिन ये रियल दक्षिणा होता है ट्रू टीचर्स का, कि जब आपके स्टूडेंट्स आपके साथ खड़े हैं, आपके साथ बात कर रहे हैं और आपका सीना जो है चौड़े कर रहे हैं, गर्व से ऊंचा कर रहे हैं और उनको कम्प्लीमेंट कर रहा है, तो मैं फील कर रहा हूँ कि मैं सक्सेसफुल हूँ कहीं ना कहीं।
सर श्योर सर, आप ही हैं सर, डेफिनेटली आप ही है सर, क्योंकि सर मैं अभी भी बार बार इन चीजों को एम्फसाइज करता हूँ सर। मुझे अभी भी याद है कि सर जब मैं आया था एन कॉलेज में फर्स्ट डे, और सेकेंड या थर्ड क्लास में आप आए थे, एंड वी वर सो कंफ्यूज सर, हमें पता नहीं था कि क्या करना है, बस इतना पता था बायोटेक्नोलॉजी में कुछ तो बूम आने वाला है, चलो करते हैं बायोटेक्नोलॉजी। हाँ बट सर एक चीज था, एक मैं सर स्टूडेंट लोगों को बोलना चाहूंगा सर, एक चीज शेयर करना चाहूंगा कि डोंट वेस्ट योर टाइम यू नो फॉर मेडिकल प्रिपरेशन, या जो बहुत लोगों ने कर दिया, आप आगे वाले जो आपके जो नेक्स्ट जनरेशन भी है, जो भी चीजें हैं, ऐसा नहीं है कि देयर इज स्कोप फॉर जस्ट ओनली फॉर मेडिकल या फिर इंजीनियरिंग एंड ऑल, ऐसा कहे, बायोटेक्नोलॉजी भी है, लोग एक्सेल कर रहे हैं और ये चीज हो रही है। इसलिए और एक ये भी रीजन है कि जैसे सब कुछ स्टूडेंट आते हैं जैसे प्लस 2 के बाद जैसे 2-3 साल मेडिकल के प्रिपरेशन में टाइम लगा देते हैं, फिर ग्रेजुएशन में आते हैं, ग्रेजुएशन होने के बाद 1-2 साल टाइम लेते हैं कि नहीं अब मैं कुछ तैयारी क्या करूं, फिर उसके बाद मास्टर्स में आते हैं, फिर टाइम लेते हैं, फिर पीएचडी। तो ये सब चीज होते होते 30 ये हो जाता है, फिर वो लोग परेशान हो जाते हैं, बोलते हैं कि नहीं अब तो जॉब ही नहीं मिल रहा है या जॉब नहीं मिलेगा, समथिंग लाइक दैट। तो ये चीजें हैं सर, थोड़ा सा हमें स्मार्टली ये करना पड़ेगा। वही मैं बोल रहा था सर, मुझे स्टिल याद है सर, जब मैं आया था एंड वी वर सो कंफ्यूज कि सर व्हाट टू डू, हाउ टू परस्यू फॉर दिस, देयर इज नो वन वाज देयर सर, आपको पता है सर, कॉलेज की कंडीशन पता थी सर। So really, I'm really thankful to you, sir, on this platform. I'm really thankful to you, and I feel sometimes privileged क्योंकि सर आपने मुझे गाइड करा, and because of that reason, you only told me, sir, कि नहीं ये बुक फॉलो करो, ब्लाइंडली फॉलो करो, ये एग्जाम है। You only told me, हमें पता भी नहीं था IT एग्जाम का एग्जाम होता है। You only told me कि IT एग्जाम का एग्जाम होता है। You only told me कि सर एग्जाम का एग्जाम होता है, ISC होता है, TFR। उस टाइम में ग्रेजुएशन में 2006-7 2009 में सर हम लोग 2009 में 2009 में वी वर द फर्स्ट कि हम लोग गए वहां पर सर के टाइम में हमारे कॉलेज से IIC और CIFR और NCBS में इंटरव्यू देने गए, एंड वी वर सो नियर, बट हाँ धीरे धीरे चीजें होती गई सर, आपका मिलता रहा हमारे हम लोग अच्छे करते रहे सर। रियली सर थैंक यू सर, थैंक यू वेरी मच
सर। एक चीज और आप ये सिलेक्शन कमेटी में भी हैं अभी लेक्चरशिप और प्रोफेसरशिप के लिए, तो बच्चों को थोड़ा सा एक ये कर दीजिए कि क्या अपने वो CV में मेंटेन करें मास्टर जो कि वहाँ पे जरूरत पड़ते हैं बच्चों को, कि जैसे रिव्यू का कितना मार्क्स है या कौन कौन सा पॉइंट वहाँ पे ऐड होता है, जस्ट ब्रीफली आप ये कर देंगे तो बच्चे वो अभी से अवेयर हो जाएंगे
सर। अभी जैसे कुछ सिचुएशन है अभी सर, अभी इंडिया में मोस्टली जो मार्क्स जो मिल रहे हैं सर, अभी तो सबने सर +2 किया हुआ है, सबने ग्रेजुएशन किया है, सबने मास्टर्स किया हुआ है, PhD का भी अभी जहाँ भी असिस्टेंट प्रोफेसर की रिक्रूटमेंट होती है सर, तो PhD का पॉइंट उसमें जुड़ता है, एंड देयर इज 10 मार्क्स फॉर दिस PhD कोर्स, सॉरी फॉर PhD। इफ यू हैव कंप्लीटेड PhD देन यू विल बी अवार्डेड विद 10 मार्क्स। तो PhD कहीं ना कहीं सर मायने रखता है। PhD कहीं ना कहीं अगर आप वाकई में असिस्टेंट प्रोफेसर को ऑप्ट करने के लिए आप कहीं अप्लाई करना चाह रहे हो, यू हैव टू हैव PhD वाला क्वालिफिकेशन, वो करना है नेट डेफिनेटली। बट अभी कुछ रेगुलेशन आए हुए हैं, 2009 के रेगुलेशंस में आए हैं कि इफ यू हैव कंप्लीटेड योर PhD विद कोर्स वर्क देन यू डोंट हैव टू, यू नीड नॉट टू क्वालिफाई फॉर नेट एंड ऑल, बट स्टिल आई वुड से सर नेट मस्ट है सर, PhD डिग्री मस्ट है सर, एंड अपार्ट फ्रॉम दैट एक सर आता है जो कि रिसर्च पेपर्स आते हैं सर। इस रिसर्च पेपर में ये डेफिनेटली वन स्टूडेंट एंटर फॉर PhD for some good institute and all, then definitely he or she will work hard and he or she will publish in few papers of in good journal। सर यहाँ पर अभी क्या हो रहा है, कुछ इंडिया में अभी ढेर सारे प्रीडेटरी जर्नल हैं, ये जर्नल कुछ इम्पैक्ट फैक्टर्स नहीं होते हैं, दे जस्ट दे डू हैव नास एंड ऑल। तो हमें वैसे जर्नल में पेपर ना पब्लिश करें और कुछ हाई रेटेड और इम्पैक्ट वाले जर्नल में अगर हम लोग पब्लिश करें तो इट्स रियली इट्स मतलब अच्छा चीज होगा। और ये यही चीज होते हैं, 3 चीज PhD, नेट एंड रिसर्च पेपर्स। इफ यू हैव पब्लिश्ड एट मिनिमम 2 रिसर्च आर्टिकल, चाहे वो फर्स्ट ऑथर हो सेकंड ऑथर with more than 2, 3, 4 impact factor or minimum 3-5 impact factor, then definitely it will be getting shortlisted for assistant professor for the interview and that. ये सब चीजें।
या राजीव जस्ट कंक्लूड, अगर किन्हीं का क्वेश्चन है तो ले लीजिए।
Yes, sir. Rajat K. Chaudhary, he's complimenting. Great session. Thank you, sir. Providing for lots of information. Then Shakshi is here. She is asking, sir, I am MSc Botany first year student. I want to ask how to start my preparation from now onwards. What steps should I follow? Can you please guide?
Yeah, so you have done your masters in botany. So you must have you must have studied all those things like biochemistry, mold biology, classical genetics, molecular genetics, cell biology, genetic engineering. Then you must have studied all those things. Then it's a positive. It's a I would say it's a positive points for you. So you already have basics. You have already foundations. You have already basic concepts. Now whatever syllabus, you just download the syllabus and whatever book I have suggested, and just be in touch with Sanjeev sir also. And whatever book we have suggested, just go through it, just work accordingly. And because you have already done your masters, so I think you must have gone through these books also, this textbook also. So refer this textbook and see which type of question they are being asked, sample question, and it will help you in through this exam.
Okay, now I think we should conclude the session. Few people are asking for your slides, so I think we can share it further.
Yeah.